வணக்கம். நம்ம இன்னைக்கு பார்க்க போகிறது ஸ்ட்ரக்சர் ஆஃப் யூகேரியோட்டிக் செல்ல இருக்க கண்டினியூஷன் தான் பார்க்க போகிறோம். வீடியோக்குள்ளே போகிறதுக்கு முன்னாடி this video is sponsored by BuyMote shopping app. BuyMote e-shopping application is one of the online shopping app. நீங்கள் உங்கள் வீட்டுக்கு தேவையான டெய்லி நீட்ஸை இந்த BuyMote e-shopping ஆப்பில் டிஸ்கவுண்ட்டோட பர்ச்சேஸ் பண்ணிக்கலாம். அது மட்டும் இல்லாமல் BuyMote வெல்கம் கிட்டுன்னு ஒரு காம்போ ஆஃபர் போயிட்டு இருக்குது. 999க்கு 25 ப்ராடக்ட்ஸ் கொடுக்குறாங்க. எனக்கு அது வர்த்தாக தோணுது. உங்களுக்கு வர்த்தாக தோணுச்சுன்னா ட்ரை பண்ணி பாருங்கள். இதில் பாப்புலரான பிராண்ட்ஸும் இருக்குது. நமக்கு தேவையான பிராண்டுக்குள்ளே போயிட்டு நம்மளுக்கு தேவையான ப்ராடக்டை டிஸ்கவுண்ட்டோட பர்ச்சேஸ் பண்ணிக்கலாம். இந்த ஆப் ப்ளே ஸ்டோர் அண்ட் ஆப் ஸ்டோரில் அவைலபிளாக இருக்குது. ஆப்போட லிங்க் கீழே டிஸ்கிரிப்ஷனில் இருக்குது. ட்ரை பண்ணி பாருங்கள். வாங்க இப்போ வீடியோக்குள்ளே போகலாம்.
biosynthetic secretory pathway abhinadana is used to move materials to lysosomes as well as from the inside of the cell to either the plasma membrane or cell engineer அப்படினு சொல்வாங்க. இந்த process எப்படினா காம்ப்ளெக்ஸா இருக்கும். The movement of proteins வந்து particular importance இருக்கும். Proteins design the cell membrane வந்து lysosomes or secretions வந்து synthesize பண்ணி ribosomesல attach பண்ணுவாங்க through endoplasmic reticulum. அதுதான் RER. Endocytic pathway na என்னன்னு பார்ப்போம். Endocytes is used to bring materials into the cell from the outside. During endocytes, a cell takes up solutes or particles by enclosing them in vesicles pitched off the plasma membrane. In most cases, the materials are delivered to the lysosomes where they are digested. Endocytes occurs regularly in all cells as mechanism for recycling molecules in the membrane. Some cells have special endocytic pathways that allow to conjugate materials outside the cell before bringing them in. Others use endophytic pathways as feeding mechanism. Many viruses and other pathogens use endocytic pathway to enter the cells. Organelle involved in genetic control of the cell nucleus. Nucleus usually prominent organelle in eukaryotic cell, discovered by Robert Brown in 1831. Considered features of eukaryotic cell in respiratory for the cell, genetic information and in control center. Nuclear membrane are delimited spherical bodies about 5 to 7 unimeter in diameter. These fibrous material called chromatin seen within the nuclear plasma of the nucleus of stained cell. This is the DNA containing a part of the nucleus. In non-dividing cells, chromatin is dispersed but in condense during cell division to become visible as chromosomes. Euchromatin is loosely organized and it contains those genes that are actively expressed. Heterochromatin is called or more tightly appears darker in electron microscope. The nucleus bounded by the nuclear envelope, a complex structure of inner and outer membrane separated by 15 to 75 perinuclear space. The envelope is continuously within the ER and several points. Its outer membrane is covered with ribosomes. A network of intermediate filaments called nuclear lamina is observed in animal cells. It lies against the inner face of the nuclear envelope and supports it. Chromatin usually is associated within the inner membrane. Many nuclear pores penetrate the envelope and each pore is formed by a fusion of the other and inner membranes. Pores about 70 nm diameter in occupy 10 to 25% of nuclear surface. A complex ring link arrangement of granular and fibrous material called the annulus is located at the edge of the H pore. Nuclear pores serve as transport routes between the nucleus and cytoplasm. Annulus regulate or aided the movement of material through the pores. Nucleus. A nucleus may contain from one to many nuclei. Nuclei is not membrane enclosed. It is a complex organelle with separate glandular and fibrin regions. It is present in non-dividing cells but frequently disappearing during mitosis. After mitosis, the nucleus reforms around the nuclear organism, a particular part of specific chromosomes. The nucleus plays a major role in ribosome synthesis. The DNA of the nuclear organism directs the production of ribosome RNA. This is RNA synthesized in a single long piece that cut from the final RNA molecules. RNA combines with the ribosomal proteins synthesized in the cytoplasm to form a partial combined ribosome subunits. The granule C nucleus or subunits. Immature ribosomal subunits then leave the nucleus permeable by way of the nuclear envelope pores and mature in the cytoplasm. Eukaryotic ribosomes. Eukaryotic ribosomes is larger than the prokaryotic 70S ribosome. It is dimer of the 60X and 40S subunit, 22 nm in diameter, a sedimentation coefficient of 80S. Eukaryotic ribosomes associated with the endoplasmic reticulum free in the cytoplasm. When bounded to the endoplasmic reticulum to form through ER, they are attached through the 60 years. Both free and ER bounded ribosome synthesize proteins. Proteins made up on the ribosomes or the RER are often secreted or instead of ER membrane as internal membrane proteins. Free membrane proteins are sites synthesis of non-secretory and non-membrane proteins. Some proteins synthesized by free ribosomes are inserted into the organelles such as nucleus, mitochondria and chloroplast. Proteins called microsamples and aid proper folding of proteins after synthesis. They also assist the transport of proteins into eukaryotic organelles such as mitochondria. Organelles in energy conservation, mitochondria and chloroplast. Mitochondria is most eukaryotic cells called the powerhouses of the cell. Metabolic process tries carbonic acid cycle and the generation of ATP, the energy currency of all life form take place there. TEM mitochondria cylindrical structures measures 0.3 to 0.1.0 by 5 to 10 UM. Some cells possess 1000 or more mitochondria. Some yeast unicellular algae, protozoa, a single giant tubular mitochondria twisted into continuous network permeating to the cytoplasm. The mitochondria is bounded by two membranes. A outer membrane separated from a inner mitochondrial membrane by a 6 to 10 nm intermembrane space. Outer mitochondrial membrane contains porins similar to the outer membrane of gram-negative bacteria. The inner membrane has special enfolding called cystine which generally increase its surface area. The shape of cystine differs in the mitochondria from various species. Fungi have plant-like laminar cristae. Eukaryote flagella are cristae shapes like disc. Tabular cristae are found in variety of eukaryotics. Amoeba can possess mitochondria with crystal in the shape of vessels. Inner membrane encloses the mitochondrial matrix and dense matrix contain ribosomes, DNA and often larger calcium phosphate granules. In many organisms, mitochondrial DNA encloses circle by DNA. Some post DNA, mitochondrial DNA is a DNA in linear. Okay, thank you.